Yes.
Okay, excellent, very good. So as I said, I'll go through a lot quickly. Do not be alarmed. You'll be spending a lot of time with dkNET over the next 6 weeks, okay? Um, so I'm almost having trouble finding where to put all my windows. I already mentioned a little bit about dkNET and what it's about. So I think we don't need to go over this very much. One thing that you will hear is FDI Labs, and that is the name of our laboratory here, the Fair Data Informatics Labs. And I've already introduced the FAIR principles, and hopefully you will see much more of those as the course goes on at UCSD, okay? So that is who we are, and this is what dkNET is about, which we've already covered. So the dkNET is a portal, as I mentioned, and it's actually divided up into different sections. There are 5 main areas. One is called resource reports. These are the core, form the core of dkNET's mission. That is, how do you help researchers find out what resources are available to them and find out about those resources themselves. So how do they perform? What can they be used for? So on and so forth. So resource reports is one main area of the portal. The second is related to this is authentication reports. And we're going to talk a little bit about that during the course, but also you'll see a demonstration of this today. And this relates to the NIH guidelines on rigor and reproducibility and the need to make sure that when you're submitting a research grant or a research paper, that you have properly authenticated the resources that you are planning to use. So if you're using a cell line, you need to tell the reviewers of the papers and grants, how do you know this is the cell line that you think it is? Obviously, a critical pillar of reproducible science is that we're actually studying the things that we say that we're studying. So we maintain a whole section that helps with understanding NIH guidelines on rigor and reproducibility, and also how you can use dkNET to help prepare an authentication plan. We have FAIR data services, and as we'll talk about in a couple of weeks, or actually on Thursday for FAIR data, I forget it's no longer a couple of weeks, it's a couple of days, we're going to talk about FAIR in the context of the open science movement, which is that it is critical for transparency, reproducibility, and really to propel data science that we start to publish our data the same way as we publish our findings that result from analyzing that data. So we have a whole section that helps researchers understand how to do that, what are the resources that are available to them, and as we'll learn about with FAIR, what sorts of standards should you be using to prepare your data so that it is findable, accessible, interoperable, and reusable. So we have a whole section on that, and that will be, I think, Thursday's class. We maintain a discovery portal, which lets you easily search across multiple databases. Over 200 of them currently. And you might say, I have Google already, what do I need dkNET for? But when dynamic databases are made available on the web, it's actually fairly difficult to search them. Google searches static web pages, but if you generate that content dynamically as the result of a query, it's considered part of the hidden web because it's not really accessible to search engines. And so dkNET maintains a portal into that so that you can issue one query and see what sort of information is available across all of these different databases. And finally, we have the Hypothesis Center. These are these powerful tools for data mining and hypothesis generation, of which our current one, the main one is SPP, and Neil's going to be talking about that later in the week. And I know that all of you have taken a look at dkNET. And that was actually one of the things in your application that we looked for to make sure that you had actually gone on and visited the site. So I know that you're familiar with a lot of this. This is just the homepage of the website. You can see it's divided into resource reports, authentication reports, and FAIR data, the discovery portal, and the Hypothesis Center. So it makes it easy to go to each one of these in particular. And also down below, we have news opportunities, right? So you can see that we have pilot programs and other sorts of things are available. So it's a good thing to visit dkNET periodically to see what is new. So I just want to talk a little bit about resource reports, and because, as I said, this is one of the main products of dkNET. How would you use it? I'm planning my experiments. I'd like to find and evaluate key resources, or I'm publishing a paper and I need to put in these research resource identifiers. So, whoops. This is what a resource report looks like. You get to search across the, antibodies, organisms, model organisms, other types of things that are available. And information that is available to us about these resources is all encapsulated into a report. So it's easy to read and easy to look at. And it gives you basic things. For example, if you're looking for antibodies, it will tell you something about the antibody and what species it's raised in, all of the sort of standard information that you get. But we've also augmented these reports with other useful information. So if other people have published using this particular antibody, we have a way of being able to link and bring you information on that. That's a very good source of information if you'd like to know how these antibodies perform, what protocol should I use with them. You can get that with the usage and citation metrics. Also, if there's any alerts that are associated with it, if something's gone wrong, if we can get that information, we make that available to you as well, okay? So it's a very useful thing to be able to explore and use, and we hope that you make a lot of use of these during your summer program and beyond. The reason that we're able to pull this information together is because of something called the Research Resource Identifier, or RRID. And I've mentioned that several times. Anita's going to mention it. You may have already come across this because a lot of journals require that you produce these RRIDs. So I just wanted to spend a few minutes on what one of these is. Research Resource Identifier is a unique identifier that is assigned to a research resource. So each antibody gets its own identifier, each model organism, each software tool. And we talked a little bit about your ORCIDs, right? ORCID is the identifier that identifies you individually as a researcher, and it is you and only you, and not somebody that might have the same name. The RRID is the equivalent for research resource identifiers. And why is this important? Well, it turned out that when people were publishing their papers, they weren't giving us enough information to know what reagent they used. They might say, I used an anti-GABA antibody, and there might be 500 anti-GABA antibodies. Just like I mentioned before, I said this is Martone, but there might be 3 or 4 Martone. So which one did you use? So these RRIDs uniquely identify those research resources that are inside of papers. But it also makes it really easy to find out who else is using these, because it's a unique string that's computer friendly. It can look at that RRID, and it doesn't matter what text is written around it. It says that is the antibody I used, that is the cell line I used. So they're able to pull all of that information into a resource report because people have put these identifiers inside their papers. So the RRID is something that dkNET introduced and is one of the most powerful tools currently for rigor and reproducibility in biomedicine. And that is the basis of these resource reports. It lets us do a lot of things about this the more RRIDs are used, okay? So you'll see this a lot. This just shows an example of one, right? RRID AB1855196 is this particular antibody. It resolves to information like here's the catalog number, here's the vendor, so on and so forth. And when people publish their papers and they put these RRIDs in them, it makes it really easy for us to pull this information in. So it's a very, very powerful, it's simple, but very, very powerful, okay? And that's been one of the core activities of dkNET over the last few years, especially Anita Bendrowski, making sure that journals ask for these. And currently over 1,000 biomedical journals use the RRIDs. So this just shows you again that CellPress, Elsevier, Wiley, Nature, those of you who are starting your careers already recognize the names of these types of journals. These are some of the top journals that are available, and they're all supporting RRIDs, okay? So very, very important tool. One of the things that this also, again, lets us do is to let people know through those ratings and alerts when something has gone wrong. So as Anita will tell you in her lecture on rigor and reproducibility, we've known for a long time that certain reagents and tools don't work the way they're supposed to in certain contexts. But even after this information was known and a paper was published about it about 10 years ago, we still have hundreds of papers that are using these particular research resources. Some of the worst are cell lines. These are tumor cell lines that have been misidentified. They've mutated. If you read the book on the immortal life of Henrietta Lacks, you know, the HeLa-cell contaminated a lot of stock. So people weren't studying what they thought that they were studying. This information was available, but it was very hard to distribute this to the researchers. So resource reports, because again, we have the RRIDs able to pull that information in, let people understand when they go to publish their paper that the thing that they're publishing on actually is a problem, right? So we have some evidence, as I think Anita will show, that when people use RRIDs, there's actually a lot fewer of these problematic resources that are being used. And we are hypothesizing, we don't know for sure, that it's because authors all have to go for most of the journals that they publish in, get the RRIDs. And when they do that, they get a big red warning sign that says, hey, this is a problem, okay? Ideally, however, we don't want people to find out about these problematic reagents after the study is done. We'd like them to find that out before the study is done. So that's why we developed authentication reports, where you can put in a list of the reagents that you use or are planning to use. It will tell you, do we have any known alerts on them? But more importantly, because you've now created a living report, if some problem arises after the fact, you can also be alerted, right? So it becomes a sort of an information network, kind of like an FDA recall on certain foods, right? Because we can track these through these numbers. You now can be notified if something comes up after the fact. And this just happened in neuroscience, where a major data release of a gigantic project on child health found an error in one of their major data releases. And quite a few papers had already been published. This is a way then to go back and say, you know, there's an error there. You're going to have to account for it. So it's a really powerful tool, and it's the basis of these authentication reports. The authentication reports, again, are based on the NIH guidelines for rigor and reproducibility, where NIH is now requiring that you give your plan for how you're going to authenticate your resources when you submit your grant. And a lot of journals will also require that you have this type of information when you publish your paper, because these research resources need to be able to perform the way that they should perform. So again, I think I skipped something there. No, I didn't. You can go to the NIDDK or dkNET website. You can learn about this, and you can learn about the tools that we have that support them. This is just an example of the type of report that you can prepare. Okay, I won't go through it now, but hopefully it's well enough documented that you'd be able to do that. Okay. We already mentioned FAIR. There's a big push in NIH and journals and just society in general. I think when we talk about this on Thursday, we saw how much during COVID it was important that information be made freely available and that the data be made available. You've already seen a couple of retractions and other things, right? Because when people are able to look at the data, they often find mistakes and errors. So it's very important that we start to learn how to make our data available. And dkNET is, again, taking a leadership role in that for DK fields, so that we teach you how to make your data fair, findable, accessible, interoperable, and reusable. And some of the most important ways of doing that is that you actually submit your data to these repositories whose job it is to make data fair. So we have a whole section of that on the portal that allows people to go and look at those. You learn about things like data management plans and others. We've gathered these resources together. So you'll hear a lot about that during the week. This is research data management, as I've mentioned before. This is very critical. How do we manage our data in the laboratory? Is very much, is extraordinarily important. It is difficult. A lot of labs are not equipped, but if you're going to share your data later on, even with your colleagues or yourself in some future time, you need to make sure that you properly document and structure your data so that it can be used. So we do have some resources on data management. NIH and both NIH and the National Science Foundation, when you put in a grant, require that you have a data management plan, and those are going to become more and more important, right, as biomedical science moves towards being a more data-driven science. So this is an important set of skills to learn in the early part of your career, because it will become increasingly important later on. I've often heard senior colleagues, myself included, say, I never ever regretted spending the time properly annotating and organizing my data. I have often regretted not doing that, right? Because it takes time and effort, but it is well worth your time and effort, and we will go through that, and dkNET also has some resources to help you with that, okay? We'll go through that later. And then finally, the discovery portal, we mentioned, again, you can issue one query like diabetes, and here you see over 200 different databases and the results that they have. If you click on them, you get a synopsis. It'll let you go and visit these databases. So it's a really powerful tool if you're trying to kind of get a sense of where data might be available or what's known in these other resources beyond the literature. You can use the dkNET discovery portal. And this is just an example right here. You have various filters and other things that you can play with. Okay. There is on dkNET something called the community resources, and that means these are general databases that are all over the place. They could be in neuroscience, they could be any place. But we do have a more focused set if you'd just like to focus on databases that are relevant to DK, you can use that filter. And here you can see some of the centers that NIH or NIDDK has funded that are meant to, again, support the research community. So you can both do a broad search and you can do a more focused search with dkNET. And finally, we have the Hypothesis Center. I know a lot of you looked at this in your applications, and many of you mentioned it already. It's an extraordinarily powerful tool, and I'm hoping that Neil has an opportunity to present some of the work that he did analyzing pathways involved in COVID. He has a paper that's coming out on that, and it showed how just using existing data, you can make very, very powerful discoveries that are clinically meaningful. So I think Neil will probably mention it. If not, you can ask him about it, because I thought it was one of the best examples that I've seen of the fact that data, when it's properly managed and it's fair and made accessible, you can do a lot more with it than just usually what the researcher did in the first place. So Hypothesis Center and SPP lets you find top genes relevant to signaling pathways. You can take gene lists and try to find out where they fall. A very, very powerful platform, and Neil will spend an entire day going through that. So I think I hadn't been noticing that COA very nicely organized these and told you when the lecture is going to be right here. So that will be on June 24th. So again, you know, just as a summary, I know that's a lot of information, and it's really the type of thing where you have to be able to explore it. But dkNET helps with resources, rigor, reproducibility, just general discovery and hypothesis generation through all of these different tools that we have available. And we really hope that you spend some time looking at these, providing us feedback. Everything can always be made better. And we welcome any criticisms, any compliments, because it's, as somebody said once, the complaining customer is your best customer. If they care enough to complain to you, they're telling you really valuable information. So please feel free to be very honest about anything that you look at. Okay. We also encourage you to stay involved with dkNET even after this program is over. We have newsletters, we have Twitter feeds, we have webinars, we have opportunities, funding opportunities. So you know, it is really a good information source to support your resource.